In our last video, we saw how the famous and incredible double helix structure of DNA was discovered through the hard work of many different scientists, especially the X-ray diffraction data obtained by Rosalind Franklin and Maurice Wilkins. As we saw, this double helix structure is made possible because of the structure of our individual nucleotides. So we have our phosphodiester's linking our sugars together. This forms our backbone, which is marked here in blue. We have our bases that are hidden on the inside that end up hydrogen bonding to each other. And with our double helical structure, we get this major groove and minor groove pattern repeating across our structure here. So this is the major groove where you see that large gap with our bases there kind of exposed between those the blue of our backbone, and our minor gap is right here. We just see that little gap there with our yellow bases exposed on the inside. So the bDNA structure of this double helix is the most common and the most important DNA structure. However, it's not the only 3D structure that our DNA can form. So this will be the most common structure. This is the structure that we typically think of when you think of DNA packed in our nucleus, but it can rearrange into other shapes in certain scenarios. So in this lecture, I want to focus on the different kinds of 3D structures that we see with DNA, but then also with RNA, because as we'll see, RNA actually can lead to a lot more interesting structures compared to our DNA. The first alternative DNA structures that I want to look at are other forms of our DNA double helix. So in the middle here, we have the normal typical B form of our DNA. So this is the standard double helix that was first proposed. And then on the left and right here, we have our A form and Z form. So as you look at these, you'll notice that they are still double helices, right? They have that same typical pattern of our sugar backbone here in blue, and they still have the same kind of general pattern of our yellow bases pointing towards the inside. But they have slightly different shapes as you compare these different helices. So we have our side view on the top. So you can see there are some differences here, especially when you look at the Z form. But then you can also see more differences as you look at these top-down views here at the bottom. Right, so you can notice how the B form, your bases are kind of all closely packed together in the middle, whereas there's a little bit of a hole here in the middle of the A form. Now again, some of the key structural features that we should note in our B form is that we have our bases here sitting essentially pointing towards the inside of our helix and resting approximately perpendicular to the axis of our helix. Again, we have those major and minor grooves that I pointed out before, where you have this kind of uneven spacing of our helical backbone. And so if we look at that and compare that to our other helices here, so you'll notice in the A form, our bases are not quite perpendicular to our helical axis. In the Z form, they are still essentially perpendicular, but you notice that the backbone here kind of zigs and zags around as opposed to just having that smooth, curved helical structure. So those are some of the most immediately obvious details between these three different forms. The differences between these forms become more apparent if we look at the key parameters for each of these different helices. So you'll notice, first of all, we actually have a different handedness to our helices. So our A form and B form are right-handed. Remember from our talk about proteins that refers to the direction of the curling of our helices. So for a right-handed helix, you would point your right hand with the thumb pointing along the direction that your helix is proceeding, and then you would curl your fingers, and that's the direction of the coiling of our helix. So the A and B form are both right-handed, whereas our Z form is a left-handed helix. So it's curling in the opposite direction from our other helices. You'll also notice a difference in diameter here. So our A form is the widest, And our Z form is the narrowest. So our B form is kind of intermediate there in terms of width. That also then correlates to a difference in our length for our helix. So the Z form as the narrowest is also the longest helix, right? It has the largest rise per base pair. And our A form as the widest is also the shortest. We only have 2.6 angstroms per base pair here. And that also correlates to a difference in our base pairs per helical turn. So you'll notice we have slight difference in the number of base pairs that we have as we complete a full turn of our helix. Again, as I mentioned before, we also have a difference in the angle of our bases. So the base tilt compared to the axis of our helix. So the closer this is to 0, the more perpendicular our bases would be compared to that helical axis. So our B and Z form are very close. They're almost perpendicular to the helix, whereas our A form is tilted 20 degrees relative to that perpendicular position. Some other important differences here. So we have a difference in the conformation of our sugar. So remember, I talked about the different possible conformations within our sugar ring, essentially the different possible chair conformations for that little 5-membered ring. And we also have a difference in the conformation of our glycosidic bond. So our glycosidic bond can be anti with the base pointing kind of away from our sugar, or it can be syn with our base kind of pointing back towards our sugar. Now our Z form is very unique here because it actually has an alternating pattern based on whether our base is a pyrimidine or a purine. So we have a C2 endo conformation for pyrimidines, and we also have an anti-conformation for pyrimidines, but then we switch both of those for our purine bases. So our purine bases have the C3 carbon endo, and they are syn, so they'd be pointing back in the same direction as our sugar. So as we return to our structural diagrams here, you can note some of those key differences in our parameters that I pointed out. So notice how short our A form DNA is compared to our B form. And again, you can see that our Z form is the longest structure here. That alternating pattern of our Z form is what ends up leading to kind of this zigzag structure for our Z form here as well. You can notice how the position of our bases is kind of changing. Some of them are much closer to the backbone. Some of them are pointed further out. Again, remember that they're changed their conformation. Some of them are anti pointing out, and some of them are syn pointing backwards. Of course, I already pointed out the key difference in the tilt of our base pairs along that helical structure. So when would we expect these alternative DNA double helices to form? As I mentioned earlier, our B form is the most common double helical structure for our DNA. So for the most part, when you're looking at DNA in a nucleus, it's usually going to be in the B form. The A and Z forms will tend to occur in more specialized scenarios. So that the A form, for example, occurs when our DNA is more dehydrated. So as we saw with our proteins, water has a significant effect on our structure. So it tends to push our hydrophobic groups to the inside of our structures so that we don't have that interaction between our water molecules and our hydrophobic groups, which creates the ordered structure of our water molecules. So in the case of DNA, we see that with our bases as the most hydrophobic portion of our DNA, hiding on the inside of our helical structures. In a dehydrated state, we would have less water molecules, which would reduce the effect of our hydrophobic effect on our overall DNA structure. And so in the A form, then our bases move a little bit, and they end up more in this wider ring on the inside of our helix. They're still kind of hiding on the inside, but they're maybe less constrained and packed together as they are in the B form. The other scenario where we tend to see our A form is with RNA. So RNA, as you might remember, has an extra hydroxy group on our sugar ring, which can change the interactions that it has. So our RNA will tend to then form this A form double helical structure, at least when we do have double-stranded RNA. RNA does tend to form a lot of other more complex structures and is often just in single strands. So we don't necessarily see it in this A form all that often. But if it does form a double helix, it will tend to be this A double helix. Our Z-form structure over here on the right will occur in scenarios where we tend to have alternating purines and pyrimidines. So it's affected directly by the sequence of our DNA, similar to how the primary sequence of a protein will affect the tertiary structure of that protein. And again, as we saw with our parameters here, our Z form is strongly affected by the pyrimidines and the purines within our sequence. So it's only if we have the right pattern of our actual bases here within our sequence that we would tend to form this Z DNA structure. So what is the relevance of these alternative DNA structures? What do they do for the actual function of our DNA? This is actually an area of active research. So we don't know for sure what these alternate DNA structures might do, but we do have some guesses as to what they might do based on some interesting experiments. In order to study the role of zDNA, a group at MIT performed an experiment where they took an affinity column and they coated the beads in here with a zDNA linker. So they had a sequence that they knew would form zDNA based on the pattern of the nucleotides within the sequence. And they coated that on their beads inside of the column, so that then when they passed a solution of proteins through, all of the proteins would elute quickly, represented by our yellow and purple beads, except for proteins that bound specifically to our zDNA. So our green proteins here are proteins that are interacting with the zDNA that's coded onto our stationary phase of those beads that are in our column. And so they would end up getting stuck in our column and could then be eluted later through adding extra zDNA. So what they saw, they took their fractions from this column and they put them in a SDS page gel, and they saw this protein pop up here, marked with our red arrow. So the numbers on the bottom are representing the different fractions from the column. So the initial fractions, you have a lot of different proteins coming out, but then only our proteins that bind to DNA, our zDNA specifically, come out in these later fractions when they're adding or altering their conditions to elute that protein that was bound to the zDNA. With further experiments, they were able to identify one of these proteins that binds to zDNA as the ADAR1 protein. So the ADAR1 protein binds specifically to zDNA, and it suggests that zDNA could have a role in controlling the function of ADAR1. And so what they suggested is that ADAR1 might actually be playing a role in the transcription process. So during transcription, we of course have to have our RNA polymerase binding to our DNA and then copying the template strand to create our mRNA. And we have many different kinds of transcription factors that can influence the binding of our RNA polymerase, either increasing or decreasing the binding there. So through this formation of a zDNA structure, we can change the interaction of our protein with our DNA, and then also change the transcription process. So in this case, they were suggesting that it would increase transcription, so it would promote the transcription there, or promote the expression of whatever protein is coded by this gene. Although you could also imagine a scenario where the protein binding might reduce the binding of our polymerase and then inhibit the expression of a given gene. With our Z-form DNA, we saw that the specific repeating structure of our nucleotide sequence would influence what 3D structure our DNA would ultimately form. We can see the same sort of scenario in many other structures as well. So here we have an example of a palindrome repeat in DNA and a mirror repeat in DNA. So this is based on the pattern of our nucleotide sequence. So in a palindrome, we have one sequence on one strand that's repeated on the opposite strand. So you'll notice these are superimposable if we rotate it around the axis of our helix first, and then if we rotate it around in the opposite direction, right? So rotate it perpendicular to our helix. A mirror repeat, as you'll see, is one that just repeats in the same strand. So we have one sequence here on the left, and then it's repeated inversely on the right. And so if we rotate this around a plane of symmetry right here, we would be able to superimpose our sequences. So one of the unique things that we see with our palindromic sequence here is that we actually have a sequence that can base pair with itself, right? So since our sequence is repeated in the other strand, that also means that the complementary sequence is repeated in the same strand. So rather than pairing with a second strand, we can actually pair between these two sequences in the same strand of our DNA. So this would be a palindromic sequence here. We have this sequence, which would be repeated on the alternate strand right here, again, meaning that our complementary sequence is right here in the same strand. So these can pair up together and form a helical structure through the base pairing in the same sequence. Again, since these would be repeated in the other strand as well, you could end up forming two of these sort of structures. So we would refer to each of these structures as hairpins because we have this tight structure on one individual strand. And when we combine these two together, we get a cruciform with this kind of a cross-shaped DNA structure. With our previous 3D structures, we saw how we could form unique 3D shapes all while maintaining the same base pairing, what we call the standard Watson-Crick base pairing. For the remaining 3D structures, we're going to look at ways that our bases combined in non-standard ways to make even more unique 3D structures. So of course, one of the things that we see when we look at our bases is we have, of course, our standard donors and acceptors that we use for our standard base pairing. So adenine has our usual donor and acceptor that pair up with the acceptor and donor of our thymine. But we also often have unused donors and acceptors around our base as well, right? So our top amine right here also has another hydrogen on the left that can act as a donor. And we have this nitrogen on our smaller 5-member ring here that can act as a hydrogen bond acceptor. So we can actually use these additional hydrogen bond donors and acceptors to form additional interactions with other bases. So here we have a thymine and adenine pairing up on one side, and again on the other side to make a TAT triple pairing of our bases here. So we're using, again, our donor and acceptor of our thymine. So we have our adenine interacting with those two different thymines. We can also do this with guanine and cytosine as well. So here we have our typical pairing, donor, acceptor, acceptor, matching up with our acceptor, donor, and our donor. And on the other side, we have a modified cytosine. So this cytosine is protonated to create an additional donor and donor, which matches up with our acceptor here on the top of our carbonyl, and again, that additional acceptor of our nitrogen on our 5-membered ring. This sort of triple base pairing we refer to as Hoogsteen base pairing. Because we're able to bring in an additional base to pair up with our other two bases, we're able to create then a triplex structure. So notice how now we can actually bring in 3 different strands of DNA, and we can pair them all together. So each of these would be pairs of 3 bases, all bonding together with this sort of Hoogsteen base pairing. So essentially now we have what could have been a double strand of DNA now paired up with an additional 3rd strand here to create a triplex instead of that original double helix. This triplex formation can actually be used to modify our gene expression. So normally our gene expression is controlled by specific regions within our DNA that we call promoters. So a promoter will be a DNA sequence upstream of the actual DNA that codes for a specific protein, the protein's gene, we would say. And that promoter sequence will interact with a variety of different protein transcription factors. So these transcription factors bind to our DNA, and then they change the expression of the associated genes. So they might increase the production of a particular protein from one specific gene. Now, again, our triplex can bond to an existing double helix in order to form that new triplex structure. So we can actually design a triplex forming oligonucleotide where we match our sequence to our specific promoter sequence, and we would include our bases in there that we need in order to form that Hoogsteen base pairing. And we can then bind that TFO, our triplex forming oligonucleotide, onto our existing promoter double helix and form a triple helix structure. And this can actually modify the binding then of our transcription factor, because again, we're changing the shape of our double helix as we're forming our triplex, and can then change the expression of our gene. So this actually has potential therapeutic roles. We could use these TFOs as drug molecules, essentially, to try to turn off or turn on specific genes within our genome. So our triplex structure took one additional DNA strand and matched up with our other 2 strands with that non-standard Hoogsteen base pairing. We can also take a 4th strand and form a quadruplex, or also called a tetraplex. Now, this is specifically called a G-Quadruplex because it only forms in G-rich regions, right? So where we have a lot of guanine bases. So as we saw with our triplex, the key is going to be using these extra hydrogen bond acceptors here. So we have that acceptor on our 5-member ring. We also have our acceptor on top, which can potentially accept multiple hydrogen bonds. And then we have our standard donors over here on the lower side of this guanine that we use to make our standard base pairing. So we can take this guanine and match it up with 2 acceptors from our adjacent guanine. And we can then take the donors of this guanine and pair them up with a 3rd guanine. And as you can see, we're just kind of pairing up around this sort of cyclic structure. So now we use these donors to pair with our acceptors up here. And we've completed the cycle using all 4 guanines to hydrogen bond to each other. So again, we have 4 different guanines, which could be from 4 different DNA strands, all coming together and base pairing in this G-quadruplex. So this sort of G-quadruplex structure can potentially form anywhere where we have a sequence with a lot of guanines together. So a repeating guanine sequence. Now I should also note we do require a metal ion here as well. So this metal ion kind of sits in this empty spot right in between all 4 of our guanines, and it will interact with the extra lone pairs and partial negative charges of our oxygen hydrogen bond acceptors here. So this really helps to stabilize and hold this G-quadruplex together. Usually potassium is used for this. Potassium really fits well into this little space here in the middle of our G-quadruplex, although other cations can also potentially be used. Our full 3D structure then will look like this, where we have our 4 different strands here marked with different colors, all kind of wound together into this quadruplex structure, right? So it's essentially almost like a tetrahelix, where similar to our triplex, we're just adding on extra strands and using those unusual base pairings between our different strands to hold it together. Now, for our G-quadruplex, we do have some additional complexity here because of the different ways that we could arrange our 4 strands together. So we could have a fully parallel G-quadruplex where all of our strands are going in the same direction, but we can also have anti-parallel G-quadruplexes where our strands are going in different directions. Now, of course, anti-parallel would be more similar to our DNA double helix, where remember those 2 strands are always going in opposite directions there. Just like our triplex structure, our G-quadruplex structure also has the potential to influence transcription and translation events as we change those protein-DNA binding interactions. So this is a study that specifically looked at translations. This was a RNA quadruplex. RNA, of course, also has guanine, so it can form these G-quadruplex structures if we have a sequence that's very rich in our guanine bases. So we're looking at one specific sequence to see what the relative translation activity from that sequence was. As you can see, our wild type sequence here in blue, that was marked as 100% activity, and then they compared how other sequences affected the translation of this particular RNA strand. So they were substituting in guanines or uridines at specific locations to slightly change up the structure of the G-quadruplex. As you can see, as they changed up the G-quadruplex structure, it had a direct effect on translation. So for example, this structure right here, right, decreased the translation to a significant extent, whereas this structure, you can see it slightly increased translation, although this wouldn't be a statistically significant difference here. So you would need further testing to actually confirm that this was an actual increase and not just an artifact from their experiment. Now, one of the very interesting things that they noticed is if they changed the sequence to remove enough guanines to prevent the G-quadruplex formation, they saw no translation. So that G-quadruplex in this scenario was actually required for translation. So presumably there must be some protein machinery that's able to recognize that G-quadruplex and then trigger the translation of this particular mRNA sequence. Now, again, in this particular example, we're looking at a change in RNA structure, changing the behavior of our nucleotide. So RNA, of course, because of the similarity between our RNA and our DNA, will tend to form a lot of different structures like our DNA. However, because of the extra hydroxy group on our RNA, we'll see some additional structures forming for our RNA compared to our DNA. So we're going to take a closer look at our RNA next to see how those structures will differ compared to our DNA structures. So first, for some general background, remember that our RNA has several different forms. So we have a messenger RNA that will be carrying our code from our DNA template. This will usually occur as a single strand. So the structure right away will be a bit different from our DNA because we don't have those 2 strands to wind together in our double helix. We also, of course, have our ribose instead of deoxyribose, which the key difference there, again, is adding that extra hydroxy group at the 2' position. That would be for our ribose compared to deoxyribose. And then we have uracil instead of thymine, which, of course, this is a relatively small difference with our thymine just having that extra methyl group. So our RNA, of course, will carry the code for creating our new proteins. Now, of course, it's not a direct one-to-one ratio of mRNA to proteins. One mRNA can actually code for more than one different protein. And in order to actually translate that, we're, of course, going to need other RNA working here. So we have transfer RNA involved in creating that protein itself. We also have the protein machinery of the ribosome involved as well. So again, as I mentioned, because our mRNA is always single-stranded, we're immediately going to see some differences in our structure there. So we do still form a helical structure, but of course we don't have our second strand to wind together to create our double helix. This helical structure forms because of the base stacking interactions between our bases. So you'll notice in this helical structure here, we have kind of all of our bases lined up right over top of each other in order to get that pi-pi interaction between our bases. If we do end up forming double-stranded RNA, this will actually form the A-form double helix, as I mentioned previously. Again, just because of the slight difference in structure with our 2' hydroxy group. So that 2' hydroxy group changes up our double helix, but it also allows for a variety of different hydrogen bonding interactions within our RNA structures. So that's where a lot of the extra complexity for our RNA structures comes in compared to our DNA structures. The extra hydroxy group, along with the single-stranded nature of our RNA, allows for an incredible array of highly complex structures for our RNA. So we can see individual features of our RNA pointed out here. So we can consider these as secondary structures of our RNA that we can form depending on our sequence. Right? So we might have just single-stranded regions, or we might have small regions that are bonded together in double helices. So this would be a hairpin double helix. We have some of that base pairing forming kind of that A-form structure here. You can have a variety of different loops. You can have bulges here where you don't have your bases pairing up. So there's no matching base for our adenine here. And so you can just have a wide variety of different structures that will be combined together to create complex tertiary 3D structures for our RNA. Here we can see an example of some of the complexity of our RNA 3D structures. So this is an RNA strand that's actually used as a component of an enzyme. So you can notice some of the different secondary structures that I pointed out on the previous slide. So we have a lot of different hairpin double helical structures. You have a variety of different loops. Here's a little bulge here with a couple of guanines that don't have pairs, and so on throughout our structure. Here's a little loop coming out from one of our strands here. And of course, we have a lot of other helices and loops here as well. You'll also notice that we have some areas with little dots between our bases instead of the normal line. So those are representing areas where we have a non-standard base pairing between our nucleotides. So our RNA also uses a lot of different base pairing interactions as compared to just the standard AT, AU, GC interactions. So here we have the acceptor and donor of our guanine pairing up with the donor and acceptor of a uracil to make this non-standard base pairing. Part of the reason why this non-standard base pairing is possible is because of the more flexible structure of our RNA, right? So in DNA, when you're packed rigidly together, you really are forced into the orientation where you can only form those specific hydrogen bonds. If you're able to move around a little bit more, then you have more opportunity to find other hydrogen bonds than you normally would with a more rigid structure, right? So normally uracil would be placed a little bit lower down in relation to our guanine here in this case, and so it wouldn't be able to hydrogen bond between these lower donor and acceptor with the acceptor donor of our guanine here. These highly complex 3D structures of our RNA will also allow for additional functions of our RNA. So RNA can actually act as enzymes, so we call these ribozymes. So here we have two different examples of complex RNA structures that will then allow them to act as catalysts in a similar way to our proteins. So on the left, we have the hammerhead enzyme, and on the right, we have an intron. Both of these are involved in catalyzing RNA cleavage reactions at specific sites. And this is generally the most common reaction that our RNA enzymes will catalyze. So it usually tends to be reactions involving other RNA molecules. So they don't have the full range of functions that a protein would, for example. As we saw with our guanine and uracil pairing, a lot of these 3D structures are possible because of these non-standard base pairs forming within our RNA structures. So here we have an example of the 2' hydroxy group of our ribose actually participating in this kind of base pairing. So we have a uracil hydrogen bonding with an additional adenine in addition to a pairing up with the standard uracil adenine base pair. On the top right here, we see an interaction between 2 guanines as well as a cytosine here. So our guanines are interacting together almost like they would in a G-Quadruplex. We have our standard base pairing happening over here with our cytosine, and we even have a phosphate involved. This would be from our phosphate backbone connecting between our ribose sugars. And of course, you can see another non-standard interaction down here between an adenine, and this is a modified guanine here. One of the things that you might notice as you look at these complex structures is that we also have a variety of different modified bases found in our RNA as well. So we had a methylguanine here in this interaction between our guanine cytosine and that phosphate. We had another modified guanine here, which of course removed what would normally be a hydrogen bond donor down here on our guanine. And so that has a key role in changing the sort of interactions that occur within our RNA as well. So our RNA will use a wide variety of different non-standard bases. And I want to just briefly look at a couple of the more common non-standard bases that we see in RNA. So inosine is sometimes found in our tRNA. So it's made by deaminating adenosine. So normally we'd have our amine group up here at the top. We replaced it with our carbonyl. So now we have an acceptor donor pattern instead of the usual donor acceptor pattern of our adenine. So this will then slightly change up what it can bond with. So it creates what we call the wobble position within our tRNA, where we have this inosine that's a little bit more flexible in what it can bond to, that will then lead to a richer genetic code overall. We also will often see pseudouridine. So this is similar to our uridine, but instead of attaching at our nitrogen down here at the bottom of our ring, we're attaching to our 5th carbon up here with our ribose. So we're attaching a different position, which then, of course, changes up our pattern of hydrogen bond donors, as well as the overall shape of our base. So now we have an acceptor, donor, acceptor pattern, but with an additional donor down here at the bottom of our structure. So both our RNA and our DNA can form a variety of more complex 3D structures by either changing up the base pairing that occurs between our bases, such as the Hoogsteen base pairing in our triplex, or just by kind of changing the general shape of our backbone with those different forms of our double helix, right? The A form or the Z form. Again, these different shapes will change our interactions both between our different nucleotide molecules. So again, we see things like ribozymes that are then catalyzing reactions of our other nucleotides. Or we can also see it changing interactions with proteins, right? So with the triplex, it can potentially change the interaction between transcription factors in our DNA and influence expression of our genes. But yet again, we see that the complexity of our 3D structure is key for controlling the complexity of interactions that will form with other biomolecules. Even something like DNA that we typically think of as always forming this DNA double helix can still slightly change its structure in order to modify those interactions that it has with other protein partners. And of course, for DNA, this is critical because understanding those small changes in structure and how that relates to our expression of our genetic code is critical for understanding how our genome relates to the overall behavior of our cells.